And it's an honor to introduce our guest for this edition of the lecture series, Michael Baudis. He is a professor of bioinformatics at the Institute of Molecular Life Sciences at the University of Zurich, and also a group leader at the Swiss Institute of Bioinformatics. He's a medical doctor by training, with an extensive career with many contributions in bioinformatics, in particular in computational oncogenomics. He is also very active in projects for facilitating access to genomics data through, for instance, the Global Alliance for Genomics Health, Global Alliance for Genomics and Health, where he is member of the steering committee and a lead of the data discovery work stream. He co-chairs the Elixir Beacon project, an API for querying genomic and phenoclinic data, which will be the topic for today's edition of the lecture series. The title of the talk is Federated Genomic Discoveries, Deploying the GA4GH Beacon Protocol. So I would like to pass the word to you, Professor Baudis, and thank you very much for joining our lecture series and for taking the time for giving this lecture.
Thank you, Luis, for this great introduction, and thank you for having me. So let's start with the slides here. Yes, Federated Genomic Discoveries deploying the GA4GH Beacon protocol, and it will have, so the presentation will have different levels of technical difficulty. So I hope there's something for everybody, but slides are shared and there will be recording, as had been said before. Yes, that was a very good introduction. I don't need much more of this. Just maybe the link to Germany and especially Heidelberg, which wasn't mentioned, that I did actually my thesis in the 90s at the DKFZ said in the group of Peter Lichter. So there is a strong connection to Heidelberg from my history and tradition there. And also there I started to work in hematology at the University Hospital and later drifted through different academic institutions to more towards the informatics side of things. And that's what the talk will be about. So the work of our group, we call ourselves Theoretical Cytogenetics and Oncogenomics. The cytogenetics clearly hinting towards the history, starting in molecular cytogenetics, CGH, and so on. But oncogenomics more pointing towards the more systemic approach to large-scale data analysis, and not so much focused on individual targets, individual genes and function genomics, but more to these large-scale analysis. And our specialty has always been, it's not only, but the focus has been always on regional copy number alterations, where over the last decades actually started to establish large collections and also then try to unravel the patterns and specific connections between co-occurring events and relations between disease entities and genomic profiling data. So that's the general introduction. Now, what we have as our major tool and resource is a database. Which was also started already a long time ago and has grown considerably. It's focused on cancer CNV data, on per sample CNV profiling data from currently 116,000 samples or so, and some 10K normal genome reference samples. And it represents probably the largest public collection of such data, completely freely accessible, CC BY, CC0 for some parts of it, and includes lots of these data types also for reference data sets like TCGA and so on, but now increasingly also other genomic variation data. Now, you will see then why I start with our resource, besides being happy to talk about in general. So this resource contains really annotations for more than 800 or about 800 different tumor types, depending on what classification system you're using. And for all these tumor types, we have then the aggregate data generated from the individual sample profiles. For instance, for CNV profiles, here you can see this would be head and neck squamous cell carcinomas, more than 2,000 samples, and so on. And so it's a resource for somebody just doing fast lookups or going more into detail by querying for specific changes here for focal deletions in the CDKN2A locus in glioma patients, and then trying to figure out, is there now other specific patterns associated with that, and so on. But you also can retrieve the raw data and do the analysis yourself through the API, and the API will be the main topic of this talk. There's metadata annotations for whatever we can get our hands on. So this is all curated data we pull from resources, from publications, from Geo and so on, and then add to the database and make it available through a standardized API, the Beacon API. We have another resource built now on the same paradigm. It's cancer cell lines. It's more focused on annotated mutation data in cancer cell lines. So it's a good resource for looking at if you're looking at your model system for your disease of interest and you're looking for cell lines, and then you can compare if individual cell lines from literature data may well serve your interests. Now, one thing I realized early on is that you can work a lot on data curation, and you can spend lots of time and you can build database, and you can proud of whatever data you have accumulated over time and the quality and how you represent it and so on. But it's never enough, right? And it's never enough because data is being produced nearly everywhere with some geographical gaps. This shows, for instance, an analysis of more than 100,000 array sets, so in nearly 180,000 or so samples where they originated from, and you can clearly see the focus on data being generated in Europe and the typical coastal areas, mostly in the United States or in East Asia, but some geographic regions are really not well represented in cancer genome analysis data. And so you see two things here. The one thing is, yes, not everything is being analyzed. Not all needs are being served. Not all data is being generated where there could be new data we haven't made good use of in analysis so far. But also that you will have data from many different places, and you have to get access to somewhere. And as I said, you cannot curate all data production in the world. So you have to find different ways. So you have to open up the data on one side, and then you have to connect it on the other side. Now, about 10 years ago, originally 2013, with the first official meeting in 2014, an organization was founded. It's called the Global Alliance for Genomics and Health. And you see here the headline on what it tries to achieve. Basically a federated ecosystem for sharing of genomic and clinical data. So no building of databases by the organization or so, but providing tools, guidelines, ethical guidelines, and knowledge about how to make this better, and also to drive, and also the reason for why we should make data more accessible. It is supported by major funding organizations, primarily in Canada, US and UK, but other contributions are then more on the interaction level and collaboration level. So this is now some slides being raison d'etre of the Global Alliance. Why do we need more genomic data? Obviously, one can find better patterns in health, disease, statistical significance, variant interpretations, diagnosis, and in the end, advance what one calls now precision medicine or personalized medicine. Now, what types of data resources are there in general, or how do people organize data beyond their individual database, in-house database and resource, thinking more about larger structures. So you can have centralized genomic knowledge bases, like ours is one, a medium-sized one, curating and putting data in a place and manage this, right? You have trusted data commons like European Genome-Phenome Archive, where people deposit data, so support then reproducible science because you can retrieve the data at some point later. There are networks, and then one can also connect data sets. Now, the data commons approach you're all familiar with, I guess, like provided by EGA, and now the federated nodes of EGA. So other database, other centers are either copying the way EGA has handled this, or directly adopting and collaborating and intersecting with EGA, which is hosted at the European Bioinformatics Institute in Kingston and in Barcelona, is that you can deposit data there in a secure environment. The problem here is shown, so the success and the problem of EGA is shown here. There are many, many data sets of different data types, not sequencing, but other data, but they are not owned by EGA, so EGA cannot give you access or combine the data, right? So EGA has no power over this. It just provides the way to access the data. But then, as it's highlighted there, and it's a slide by Jordi Ramla, a technical lead of EGA, there are here at this time point 2,300 data access committees. So that's, yes, data is there, but we need better ways to handle this. Other example I just mentioned briefly, how it's done in Switzerland, but that's example that you see in many national systems is secure connecting nodes and so on, and then putting everything into this system. So at least we have a connection between different players, but still it's then a very protected and shielded system. So the main keyword or the main target for which sees the GA4GH for a solution is here to do data federation. So linkage of distributed data sets. There have been several publications about this. There was a special issue of Cell Genomics 2 years ago, so you can read up on that. As I said, slides are online, not going in detail, but you say data sharing, federated data, and then all the problems associated with that, trying to solve them. What does it need? So federation is a mix. You sometimes, yes, you download data, but in principle, it means first you have to identify data in different places and then make use of the information you get there. So more visiting data than data copying. However, for that you need standards. So what GA4GH is at the end is mostly a standard defining organization for all the parts of the chain, from the data donor, the patient or individual, to the user, and then again to the individuals benefiting from the generated knowledge. And this concerns all the different areas, from technical sequencing information, but also consent information and so on, and how data access committees can be made better, more digital, and more approachable. Now, this is organized in the Global Alliance in different work streams. Anybody can contribute and collaborate there. And there, which you see the different areas: data security, discovery, cloud, and so on, regulatory and ethics. And as was mentioned by Luis in the introduction, I am the co-chair of the discovery work stream, where one of our main products in the Global Alliance is the Beacon protocol. You see also many other standards there, like all these file standards for genomic data and so on. BAM, CRAM, VCF, and so on are also now part of GA4GH developments and widely adopted by large organizations. Which brings us to the Beacon protocol. So federating genomic discoveries. Beacon protocol was a very smart idea when GA4GH was kind of initiated, and the idea was to really ask data producers, contributors, if they can open up their data stores, which frequently were large VCF files or so, to these simple queries. Just say, allow a query wherever you are, allow just this query: do you have this genomic variation in your database? And the idea was to get people engaged, get people excited, to have some use cases with real data, and therefore then starting a development process for improving this. Sorry, there is noise in the background. I hope it doesn't go too long. Lots of the principles we will come to later have been already then outlined by Jim Ostell from NCBI, who presented this idea in 2014, especially then that there should be a version 2, which then goes beyond that, not just this variant matching and variant discovery, but providing more utility. So Beacon V1, actually the real final version was only in 2016, but before there were 0 point, the usual 0 point, and so on, working, testing versions already with large adoption, allows these queries, and these queries can run over an aggregator against network, not a network, against many nodes linked to the aggregator, which just queries all these nodes and gives you then answer about this variant's existence, for instance, for a rare disease variant in different institutions. Here's a timeline already of the development. You saw that 2014, original idea, and then it went to version 1, improved, went to, over the years was improved, and also the network implementation and so on. And already in 2016, we started, or even a bit before that, to work on the version 2, which should have different features. And the different features here are presented, also a slide adopted from Jordi. It's basically that we would have a data model representing clinically or biomedical research relevant domains, entities, to be queried and to be represented and to be delivered over the Beacon framework. So the framework is the technical part handling the data delivery, and the data model then are these entities, basically your databases, storing information about genomic variations, biosample data, or information about individuals like sex and age and history and so on. So Beacon V2 focused more towards the clinical part. So what Beacon V2 now can do is you can run queries using Beacon V2, which go like that. Can you provide data about focal deletions in the CDKN2A gene locus in glioblastomas from young patients? And these should have unrestricted data access, maybe because they are anonymized or so on, because I want to use the data. And this can be done now, again, in this federated manner to all the nodes supporting this. And Beacon was approved as an official GA4GH standard in April 2022 and adopted by quite a number of organizations. Now let's go a bit more to details. So Beacon, what is a beacon? Beacon, large, right, is the protocol API with containing framework description and the default model, basically the schemas for all of that. The Beacon, a beacon is an implementation of that, uses the framework model and its own iteration. Then you can have beacon aggregators. A beacon aggregator is just a mechanism which has a front end for input of queries, and then has a list of beacons. They don't have to know about the aggregator, it just has to have access to them. And then you can federate out, it can send these queries to many of these individual nodes and retrieve the results and then give you and combine then the results. Now, one can this also make even a bit more advanced, and advanced version is then that you have a network. A beacon network is usually considered something where there's some control over the interaction between nodes and individual and the aggregator combining, assembling this network. So beacon networks are frequently also built in a more controlled context, that is between collaborating institutions, which don't also use the same type of parameters and so on. So you have a more harmonized approach to that. This can also be behind a firewall, or like in something like a protected network. So it doesn't have to be public. Yeah. And it can expose, I will mention this later, a public part, which may then be more limited than the internal part. How does this look now? The Beacon default model, that's what we saw. We saw individuals, biosamples, runs, and so on. And that's basically the schema, how these different entities are connected. So mention again, Beacon framework, very open, allows many models, not only the default model, but for making use of Beacon, that is varying many different beacons, you want beacons to support the same model, right? To be able to combine these queries. But in principle, Beacon framework can also then be used for other models. So if you implement a framework, you have leeway to do more with different types of data. So adherence to a shared model empowers federation, but the use of the framework then also facilitates general adoption of the ecosystem. Now here I will run through some examples. Now you saw the first query type, just this Venn variant. Now here are different types of genomic queries being supported right now. For instance, we don't define so far, this is changing right now, specific query types, but one can use combinations of different parameters now. So one can do typical sequence queries as shown before. One can do now range queries, that is, if you don't really know what you want, what you will find, but you are, for instance, interested in all biosamples having a variant in a given gene or in a given region of a gene or in the promoter region of a gene, you can do a range query, and you may then limit this with variant types and so on. But you can be a bit more fuzzy about this. You can say, give me everything hitting TP53 or so, and then get information about or get the variant spec matched with these queries. One can do structural variant queries, or which here is an example of a technical, it's called a bracket query. It's used, for instance, to find CNVs, copy number variants covering a whole gene, can be specified with these bracket queries, which have two start and two end positions, and then type and so on. And you can change the size and position. So that's very flexible. So any contiguous region on the genome can be matched with these types of queries. The important point really powering the biomedical, also the technical query part of Beacon are filters, which is a very simple concept, just using queries or query objects and post requests. Don't have to go to this right now, but so you have to harmonize your data using sets of identifiers, and then your Beacon can tell also the requesting client which values you have in principle available in your Beacon backend, and then one can use this to construct queries. And there are fancy things like like term expansion demonstrated here in our implementation, actually, where, for instance, a search for some higher level term is then expanded to all the child terms representing subparts of this concept. So using a lot these modern hierarchical ontologies. And so, but one can also define one's own filter for their own resource and implement then custom paradigms of that. How to implement a Beacon? Yes, there are different ways. I mean, it's just code, right? And there are schemas and descriptions and so on. You can just do standard web stacks, databases, API, and so on. There is a reference implementation provided by the Barcelona people, and we have our own stack and Beacon implementation, which has a bit more functionality compared to that. If you come to networks, it's important that we are working basically towards that Beacon network requests and responses in their evolution currently will basically be the same as individual Beacon responses. I don't want to go into that detail. Now, not finished yet, but just shout out to many, many people involved with that from different areas of GA4GH and especially of Elixir, a European bioinformatics organization supporting our project development. Now, just briefly, I mentioned at the beginning that our databases use these open API for data delivery, and that's basically because all the progenetics and cancer and so on databases, they run completely on the Beacon API. And progenetics had been one of the reference test resources during the approval process. How does this look like? Well, we have it's just code. We implemented our own, it's called a Beacon package and some helper packages. which basically mimics the Beacon data model, the entities in its collections in a non-SQL storage and object databases on MongoDB, and then uses Python package and then a standard web codes, plus some support collections for gene positions and so on. And additionally to the standard Beacon protocol, we always have had a Beacon Plus version. We always push a step ahead for testing what else could be done in Beacon. So we also support things like geographic queries and so on, which is not directly part of the Beacon data model, or also delivery of Phenome packets, another GA4GH standard, which is also not part of a standard Beacon response. All the code is available on GitHub and CC0, right? You can download, play, and give feedback. That's important. There's also an R API, obviously called PGX RPI, for accessing data of our resources, but it is being morphed into a general Beacon API for R. It's developed by Hangjia Zhao in my group. Now, briefly at the end, genomic data and privacy. So Beacon, yes, Beacon, is it safe? So the original query was thought to be, very privacy protecting, and that's why it found so widespread adoption, because you just could query against these variants even in aggregate data. But immediately, and we knew this when starting the Beacon development, that this can be if you ask just enough questions, you can re-identify. If you have a genome, you can take the variants from the genome, take the rare SNPs, and then run the query against these aggregated collection, even so till you have a statistical match that the individual exists in this collection. There was an example, a fast publication from a group at Stanford directly following the Beacon development. Now, in Beacon v2, anyway, we break this relative security by design concept, because we also allow data delivery, and we also allow many more query options, so you can do much more informative queries. Beacon doesn't itself, the protocol doesn't itself has security baked in, but it assumes certain levels. So it has several levels of data delivery, which then can be mapped to these to concepts like authorization levels. And the granularity levels, as they are called in Beacon, they are Boolean. So you just give an answer yes or no. Count, you just say how many, or record level, where you can deliver data and so on. And this can be mapped to authentication practices. And here's just some of the last slides. There's just an example which we are just currently implementing, or better collaborators at the ETH, Scientific IT Services, implement currently a secure access procedure which combines our Beacon backend running data in a secure enclave there with then authorization servers which allow then to gatekeep what's going in and out. And so they have the Beacon, the implementation of the Beacon API. You have a gateway which can reside in a virtual machine in between. And this can then just hand over, tell, okay, here's an authorized user, and that's the name of the user. And then you can match the name of the user against your own maybe permissions in your Beacon. If you say, oh, yes, I have some, which I only allow some users specifically access, even if they're authorized. And you can use the gatekeeper also to clean out responses which may have too much data. Yeah, you can say, okay, this is a public request, so I only allow Boolean yes or no responses. Everything else is being stripped. And this is I think a kind of powerful security concept to do that. So what can you do as somebody interested in genomic and phenoclinical data and access to such data combined with genomic data? The important point is that whatever you're doing, whatever data you're producing, your user will support fair data principles. So the discovery of the data, access to the data is at a different level, but in principle, make the data as secure as necessary, but as open as possible, according to best open research data practices. And get involved with data standards development so that you yourself in the end can be better served for that. One way to get involved is with GA4GH. This is now a technical meeting we will have in April. We will have it this time in Switzerland, actually, in Ascona. This is for invited guests, but one can also register online for Zoom participation. But and as you adjust, this is a snapshot from the schedule, and you can see Beacon is kind of a topic popping up in many of the discussion sessions and working sessions. And what are we working on right now? We are working on making Beacon a bit more powerful. As always, there's development going on. For instance, defining other ways to query based on user feedback, for instance, cytogenetic queries, functional elements, and so on. And there we invite actually people, and you can contact me if you want to get involved into such use cases for that, which you don't see supported yet, then you can get invited to the Scouts team. So Internet of Genomics, well, it won't be there that easily, but we're working at least to make data a little bit more discoverable and a little bit more usable to users in different places. And this talk came from Switzerland, as is obvious from this photo. Thank you.